Good evening, everyone. I think we'll get started, so may I have your attention, please? Very quick introduction. My name is Peter Wu. I'm one of the gastroenterologists based in St. George Hospital. I'm a senior lecturer at UNSW. Before we get started, I would like to acknowledge the Bidjigal people that are traditional custodians of this land. And I would also like to pay respect to the elders, both past and present. Now, welcome everyone to our public lecture this evening, Gut Feelings. I'm exploring the role of the microbiome in digestive disorders. Now, this evening's lecture is brought to you by ANGMA. Now, a very brief history about ANGMA. ANGMA is an organization that comprises lots of Australian scientists and clinicians who share a common interest in gut health, specifically how the connection between our nervous system and our gastrointestinal tract. We meet once a year to have to share the scientific advance, and we also like to bring forward a public lecture. And this is a way for us to engage our community and share what we have learned. Now, the theme tonight is microbiome, which seems to be a buzzword for a lot of things that you read in media. If you already know a little bit about microbiome and want to learn more, fantastic. If you don't know anything about it, even better. This is a perfect forum. This evening, we're going to hear from three very exceptional speakers. Professor Ahmed El-Omar, he's from St. George Hospital, UNSW. Professor Sunny Wong, he's one of our invited speakers. He traveled all the way from Singapore, only just landed this afternoon. He's a great friend of mine. And then we have Professor Nick Talley from Newcastle. Now, I very carefully chose this list of speakers for three different reasons. Now, they are all world-class clinician researchers that have accomplished so much and contributed immeasurably to the body of knowledge in this topic. They're all fantastic speakers. And lastly, I'm a huge fan of every one of them, and I'm going to tell you why. A bit of housekeeping first. We will reserve the Q&A time to the very end of the public lecture. There are a few different ways that you can ask questions. There will be microphones walking up and down. On either side, you can just raise your hand and we will bring the microphone to you. If you don't feel like using the microphone, you can please scan this QR code and type in your question there. I will be monitoring the chat group during the lecture, and I'll be curating the questions to our panels. And lastly, if by all means, at the end of the lecture, come down here, have a chat with us. Now, it is important that we do not see any of the advice here as a medical advice. And if you have any specific health concerns, you should always talk to your own doctor. Now, our first speaker is Professor Ahmed El-Omar. Ahmed had joined us at St. George Hospital in UNSW in 2017 after he became one of the world-class gastroenterologist and researcher. After he came here, he quickly established our first microbiome research center in Australia. Currently, he has at least 100 active research projects worldwide with collaboration with a lot of different parts of the world. Now, I will not have enough time to list all his accomplishments, but I think this is something that I really have a great amount of respect for, and that is he's the editor-in-chief of one of our most prestigious medical journal in gastroenterology. Now, to give you some idea how prestigious this journal is, for someone who embraces and actually defines mediocrity like myself, the lifetime chance of my lifetime chance of being able to publish in this journal swings between 0 to 1. And I'll let you guess how many times I had published in Gut. Now, despite being very busy, Ahmed had always been very generous with his teaching and mentoring. In fact, he had been a personal mentor of mine. So I'm very grateful that he accepted our invitation tonight to speak to us. Now, without further ado, please join me in welcoming Ahmed.
So thank you much, Vita, for this very kind introduction, which I don't deserve, but I'm grateful to for your comments, and thank you all for turning up on such a wet evening. We normally submit forms to make sure that the weather is perfect, but somehow there was a mistake there. We didn't submit enough forms. Now, the topic here is actually quite interesting, and it's one that most people have heard about, the so-called microbiome, which is a surrogate for gut health. And everybody knows about gut health. Everybody knows how relevant that is to our health generally. Now, if I show you this picture, this is Hippocrates. Hippocrates is the father of medicine, and he said that all disease begins in the gut. He wasn't meaning my journal, but just gut generally. And indeed, in fact, he was proven right, because if you think about this microbiome revolution, more and more, the more and more we find, we link the gut with every other organ, the gut-brain axis, the gut-lung axis, the gut cardiovascular system. It's really, really quite exciting. So this is why it's become such a topical issue. Now, does anybody recognize the statement here, the answer to the ultimate question of life, the universe, and everything? Anyone? 42, well done, yeah. But it's the wrong answer, because it's actually the microbiome. Right. So assuming that is the case, this is why actually, we were very, very keen to establish a dedicated center that does all types of microbiome research, from very basic all the way to translational clinical trials. And our center was set up in 2017 with support from various parties, including the government, both federal and state, as well as the universities, of course, local health district, and philanthropy. So we're very grateful that we established that. And the purpose wasn't just to have a building or a bunch of labs. It was to actually do research and inspire the next generation of researchers. And this is why when we set it up, we were actually very, very cognizant of the healthcare priorities of the Australian government and tried to mirror that in the themes that we've established: cancer, women's and children's health, infection, inflammation, immunity, which covers a huge amount of medical problems, mental health and neuroscience, and critical care. And as Peter mentioned, over the last 7 years, we've been very, very busy and established collaborations with almost every institution, every university, not just in Australia, but across the globe, which keeps us very busy and excited as well. I think this is really wonderful. Now, the microbiome. So you will have heard about the microbiome, and you will also heard about the term microbiota, and it's a little bit confusing. So microbiota is just the bugs. It's whether it's bacteria, viruses, fungi, archaea, all of these things are just the microbiota. But collectively, they have obviously quite a powerful genetic component, and that's the microbiome. It's the genetic, the collective genomes of all of these microbiota, and probably what they produce, because ultimately the genes produce something, and that's basically what makes up the microbiome. Now, it might surprise you that we're mostly microbial. There are more microbial cells in the body than human cells, and the estimate is 1.3 to 1, but I'm convinced that it's a lot more, because this doesn't count viruses and other little things that we probably won't be able to quantitate. So that's the number of cells. However, if you look at the genetic power, it's massive. It's 200 to 1. So all of these microorganisms are producing things that impact on our health. That's why it's so relevant to human health. It weighs around 1.2 to 2 kilograms, and most of it is in the gut. So the poo that you have in your gut has probably half of it is actually made of microbiome. Some people have a lot of microbiome, and they tend to kind of gravitate towards certain professions like politics and other things. So they're really full of microbiome. Now, in terms of the metabolic output, that's equivalent to the liver. So this other organ that you're carrying around in your gut and elsewhere in the body produces so many metabolites as much as the liver. Liver is your industrialized zone that actually does a lot of things for your body, whether it's producing things or detoxifying things and so forth. So it's really quite relevant. That's why it's important for human health. Now, in terms of how we get it and how it stabilizes, I'll talk about this in the next few slides. But generally speaking, you start with very little when you're born, and then you stabilize, you get your kind of future complement by the age of 3. That's why that period is very, very critical. So the take-home message is that this microbiome is a new frontier in medicine and promises major advances in the near future. Already, actually, but just in the near future, we'll get a lot more. This is just a little video. I hope to kind of speed up a little bit. It was given to me in confidence by Professor Shinji Fukuda from Japan, and it takes you through a journey from the mouth all the way to the, to the colon, and just shows you that, journey of the bugs. If you swallow any bugs, it just goes through your gullet, your esophagus, and then it enters this big sac called the stomach. And the stomach is very important because it produces stomach acid. And acid is there not just to help you digest food, you can actually do without it in terms of digestion, but it's your first line of defense. It's the thing that kills all those bugs. The pH in the stomach is very low, and that's what kills the bugs. So it sterilizes anything that might be pathogenic, something that might cause you infections and so forth. So this is important, and I'll mention it later, what happens if you lose your acid and so forth. But these bugs are there. They're in an ecosystem. Just imagine yourself a marine biologist, and you're swimming around with all of these different kind of, life forms and other. Now, the idea there is that you start building up this complement of that ecosystem, and for it to be successful, you've got to have, obviously, something to feed on. So most of these microorganisms feed on what we give them, and that's mostly diet-derived. So when we eat something like vegetables and fruit, and we can't digest plant cell walls, and the roughage and the fiber that we dump in the large bowel then gets metabolized by some of those microorganisms. They break them down, they produce some substances or metabolites that are very, very useful for our health. And you'll see in a minute in this video, this action of those microbiota. They're digesting away, they're metabolizing, they're producing things like what we call short-chain fatty acids. These are little metabolites that feed our colon, feed our cells, regulate our immune system, give us resilience, fight off infections, and so forth. And most importantly, keep out other pathogenic microorganisms. So you want this diversity in the bowel because it gives you that resilience to be able to fight off any other nasty bugs, but also derive maximal benefit. What you see here is now them kind of working away on those fiber and trying to generate those short-chain fatty acids. And those little clouds are the metabolites. And these could be things that are relevant to your immune system. They could be neurotransmitters. They could be byproducts of metabolism and so forth. A variety of different metabolites that are relevant to our health. I'll just skip that. Now, I talked about the gut microbiome. This is what's in your colon largely. But another very important site, and this is emerging as one of those kind of key niches for the microbiome, is the oral cavity, your mouth. That's the second most diverse after the colon. And so it makes sense because everything, you're eating everything, you're swallowing everything, even if you breathe and you cough and whatever's in your lungs comes out in your mouth and you swallow it. So that oral microbiome is actually very relevant, and it's been shown to be incredibly important for things like heart disease, inflammation. It's periodontitis is one of those conditions where you get like gum infection and inflammation, has been linked with a variety of different conditions. So it's also very important worthy of investigations. Now, big question, where does the microbiome come from? So we assume that the in utero environment is sterile, so baby's growing there. There's some debate about whether there's a little bit of exposure to some kind of microbiology, but whatever it is, baby emerges into this kind of new world and immediately gets encounters with microbiology, with microorganisms. Whether it's a normal vaginal delivery or cesarean section, that determines that first exposure. If the normal kind of pathway for birth, you get a lot of lactobacilli. These are healthy microbiota that are supposedly good for the health of the baby. If they're cesarean section, they get more of the skin microbiota, but it's not a life sentence. You could always change your microbiome if you're pursuing healthy habits. But mom is very important. So that the take-home message from here is that the mother, and this is a study that looked at that, so it looked at mothers and their babies and tracked 4 different types of microbiota and showed that the majority of the baby's microbiota comes actually from mom and mostly from mom's gut microbiome. And this is very relevant and very important because we know that mothers that have certain conditions, for example, mothers with inflammatory bowel disease, they pass on that microbiome to the babies. And if you look at the baby's microbiome and you use animal models to study the mechanisms of what they do, they tend to kind of transfer those kind of adaptive immune problems that might be relevant to inflammatory bowel disease. So it's very important. Take-home message is that mom's microbiome is probably the most relevant. After that, it becomes all about cohabitation. So whoever else is in the household, father, brothers, sisters, visitors and so forth, pets, these are all kind of contribute to that complement of microbiome that the baby accumulates. Now, this is probably the most important slide because it's the essence of the microbiome field. This interface between the outside world and our own inside world and our immune system. So this is the kind of what we call the mucosal barrier. This is, for example, in the intestines. So what's in the inside the intestines, the lumen of the intestines, is actually still outside world. And across that mucus layer, sugary kind of layer, lies where the microbiome sits. And underneath that is your immune system. And basically, you're scanning, are these bad or good? And having a very healthy and diverse microbiome, make sure that balance is correct. So you don't overreact if there's something that is actually beneficial. Equally, you have to have a robust immune response to something that is invasive, like a pathogenic bacteria and so forth. So this is the intestinal mucosal barrier. And if it's disrupted, for example, because you've got the wrong mix of microbiology or microbiota, then problems can happen. And this is demonstrated in this slide here. So basically, if, for example, you end up with, you're on a poor diet and you're growing more of the nasty bugs, and these are more inflammatory and they cause, erosion of that, gut barrier, then there's a problem, because what tends to happen is that these nasty bugs, as well as their metabolites, can translocate from one compartment to another. For example, go from the gut to the blood, and they can end up elsewhere. So that's the concept of translocation, moving from one compartment to another. And that's how something that is in the gut could be relevant to your general health, because it goes away, circulates across the whole body. Now, lots of causes for this, obviously diet, stress, both physical and psychological, can actually change your microbiome in a negative direction. Burns, alcohol infections, and so forth. All of these are powerful changes of that barrier and causing a leaky gut. That's the whole idea. Now, this is another very important concept, and that's the concept of diversity. So in the gut, you want a very diverse microbiome. Why? Because diversity gives you resilience, gives you strength, because each one of those tribes living in that environment has a particular skill. Some of them are good at producing antimicrobial peptides. They produce things that kill other nasty bugs. Some produce, short-chain fatty acids. Some very useful kind of neurotransmitters. So that mix of skill comes from diversity, and that's an index of health. So if you analyze your stool sample and you have high diversity, that usually correlates with health, and the opposite is true. So if there's reduced diversity, that means disease most of the time. So there's always that kind of balance between the good guys and the bad guys, and if you've got more of the bad guys, you've got this word we use here called dysbiosis, not a perfect word, but it just does the job. That's more of the pro-inflammatory nasty bugs, and you change from a microbiome to a pathobiome. Patho means bad. I mean, it's something obviously negative. And it's the eternal question, the chicken and the egg. The bottom line, it doesn't really matter, but if you get chronic inflammation for whatever reason, maybe because of dysbiosis, that's the root of all evil. All human disease has chronic inflammation at the heart of it, and this is a very, very important concept. Now, just this slide, just to remind you that why bugs in the gut, are relevant, and why are they relevant to something like your immune system or whatever it is. It's all because of the production of metabolites. So metabolites are chemicals. These chemicals can then, this is the currency of communication in the body. A bug by itself is, it's nothing, but it's what it produces that then can sit on receptors and then activate certain pathways or prevent something. So, and that explains why you can have a different mix of bugs, but as long as they're producing similar metabolites, so we end up, being basically united in biochemistry. This is a term that my colleague, coined a few years ago, which is we're all united in biochemistry. Mark Morrison from Brisbane, and that makes sense. In other words, it's all about those metabolites that are relevant to our health. Okay, what factors can impact on the microbiome? We mentioned diet, geography, where you live. And that's very important because that's where the soil, where you grow your food is relevant. That's how it can change the microbiome just by virtue of being in a different environment. So if you looked at a very large geographic area, you'll find variations in the microbiome composition which are just dependent on the geography. In other words, certain types of geography there that can encourage the growth of certain type of microbiota. But there is, remember I said it's the metabolites ultimately that unites us all. So we can have different composition, but the same metabolites. Many other things that impact on that. Obviously, the mode of birth, the breastfeeding versus bottle feeding, lifestyle, and so forth. And I'll, I always get asked this question: what about our genes? Are we kind of genetically programmed to have a particular complement of bugs. Is that relevant or not? It turns out that, in fact, it's not. It's just about 6%. It's been estimated. So it's much more important about the environment and much more important about the cohabitation. And that gives us hope, because if you're stuck, if you're stuck with your genes, you can't change them, and your microbiome is determined by your genes, you're stuffed, essentially. You can't change anything. So that's actually a very good point to take home. And if we start looking at diet, that's the most powerful determinant of your microbiome. And if you look at this very beautiful picture on the left-hand side here, this is a plant-based diet. All of these things are healthy. You just smile when you see that picture because it's just so beautiful and colorful and full of healthiness and so forth. In contrast, this is the kind of typical Western diet, which is very rich in meat and fat and so forth. And this is really bad for the microbiome. So this is just the balance between things. So diet is very important, and this is borne out by this very nice study that was published a few years ago. In fact, now this is an old slide. But basically, if you look at a population that takes processed animal-based food and so forth, the characteristics of that microbiome are more of that inflammation that I mentioned, in contrast to the plant-based diet, which is the opposite. It's something that actually gives you a production of these metabolites that are good for your health. The other thing, which is underestimated yet very, very important, is medications. So we're all taking medications for one reason or another. If you fall ill, if you have an infection and you take antibiotics and so forth, these have a very profound effect on the microbiome because they can kill some bugs and they can kill them differentially. So they can knock out more of your good ones versus the bad ones. And this has actually been, this is a very nice review. If you're interested, this is a very comprehensive one. But this is one of those key papers that get published. This was published actually in Nature, one of the top journals. And it looked at more than 1,000 medications that are not antibiotics. These are things that you take for high blood pressure or, conditions that are completely unrelated to having an infection. And they found that a quarter, 25% of those medications kill some of your commensal microbiota. So they have antibiotic properties without us knowing anything about it. And the three groups of medications that emerge as the top ones in terms of their damage to our microbiome are the antipsychotic medications, the proton pump inhibitors. These are the acid inhibitors. Remember what I said at the beginning about the acid, if you knock off your acid, and morphine, painkillers. So the opioid problems that you've heard about in the US and so forth, just a vicious cycle. It just keeps screwing up your microbiome. You feel more pain, you become more dependent on these medications and so forth. So this is a very important concept. Medication, I think, is very important. Now, I mentioned this because obviously the theme of this conference and the lectures is all about the gut-brain connection. And just to say that everybody knows that if your gut is playing up, you feel miserable. You're kind of depressed and anxious and everything else. And the opposite way as well. If you've got a neurological problem, your gut suffers as well. So there's this kind of bi-directional impact of both the gut and the brain. And the reason is very simple. It's because the gut is the largest nervous organ outside of the brain. It is so heavily innervated. It's the largest, kind of nervous system, as I said, it's the largest immune system. It's the largest endocrine system. It's really quite amazing how, that's why Hippocrates was right. It was just integrated so well. And it's connected directly with the brain through nerves, like the vagus nerve, but also through these metabolites and other things that can circulate. So, producing, for example, inflammatory markers or cytokines, we call them, or maybe neurotransmitters that are, from the byproduct of these things that happen in the gut. So there's at least 4 different mechanisms by which the gut communicates with the brain, and this is very relevant to human health and disease. Now, let's just look on a positive side. So there's been lots of associations between the gut microbiome and human health. Almost every condition has now been studied, and there's, if you've got a particular disease, that your microbiome is going to be different. Chicken and egg, we're working out the mechanisms, associations are not causation, and so forth. But that's where the bulk of the work that we're doing in centers like ours and across the world, trying to figure out the exact mechanisms. But the good news is that, unlike our genes, you can actually change your microbiome. And this is incredibly important because if we discover that there is a particular signature that can predict, say, preeclampsia or gestational diabetes or risk of high blood pressure, and that you can manipulate your microbiome to prevent that condition or even to treat it, that's a huge advance in medicine. That's the revolution that is happening. We could do the same with the genes, and we've had the genetic revolution, and you can have a predictive genetic risk profile. But what can you do about it? You can't really, other than just know that you're at increased risk. This is something that you can actually, it can impact on management. And there are so many different ways of manipulating the microbiome: diet, exercise, all of these things I'll mention individually now. So again, do you not feel happy looking at this? It really is just such a beautiful, wonderful, diverse, plant-based, but it's kind of Mediterranean type diet. There's fresh fruits and vegetables. There's seafood, fish, and salmon, and so forth, as well as nuts and legumes. This is really the essence of a healthy living. Okay, this is the healthiest diet. Forget all of the fads, forget all of the thing. The more plant-based things that you incorporate into your diet, I'm not saying become a vegan or a vegetarian, but just the more of these plant-based components, the healthier you are. And the magic figure is not 42, it's 30. There's nothing magic about it, it's just the more the better. And from the studies that have been done, 30 emerges as a decent number per week. So if you've got a variety of different things per week, up to 30, you're doing really, really well. Exercise. Exercise doesn't mean running marathons, it just means physical activity. It means getting off your backside and doing something, and that actually positively impacts on your microbiome as well. So it changes your microbiome just by exercising, because you generate, you activate the right pathways that kind of changes the microbiome towards a healthy one. So that's another way that you can do, healthy diet, healthy physical activity. Now, a lot of people ask about probiotics and prebiotics. The thing about these things is that the concept is very valid. So there is something called a probiotic, which is usually a bacteria that impact, have a positive health impact because they're probably producing the right metabolites and so forth. That's a great concept, and it's absolutely valid. And the prebiotics are the feed. This is what they munch away on. So prebiotics, probiotics. The only thing is that what you're, buying off the counter at Woolies and other, supermarkets is not necessarily the answer. It's much more likely that, because they've never been tested properly in clinical trials. So we have no evidence that, if they're tested and they survive, I'll be the first to take them. But what we know for sure is that if you take those natural probiotics in natural foods, fermented foods, things that are actually where it's generated in a natural, unprocessed way, these are definitely very healthy and very useful. So that's something else that you can add to that healthy lifestyle. You will have heard a lot about this thing called fecal microbiota transplant, or poo transplants. I'm sure everybody will have heard about them. Sounds very disgusting. It works amazingly successfully in one condition called C. difficile infection. So that's an infection which people get if you're on antibiotics, particularly the elderly and so forth, and it can be quite serious. And you can cure it with getting somebody else's healthy poo. Administered usually either through the tail end or the top end, either way, it works amazingly, almost 99% successful. Now, if you want to extend that to another, like a wider use, for example, to treat inflammatory bowel disease or to treat, say, diabetes or obesity, that's a big jump because those conditions are much more complex. It's not just a single infection that is, proliferating. It's a lot more complex. And for that reason, I think it's telling us something, but that something is not 100% well defined. That's why there's a lot of research on FMT and how to administer it. And it may be that we won't need to give poo, but need to give a consortia of those microbiota that is distilled and delivered in a much more palatable way. Now, these are the so-called capsules, and these are actually available. And, in fact, some of them are even what we call lyophilized. So this is the poo is actually, frozen and dried, and it looks exactly like a capsule. And it works for C. diff, but there's a lot of work that needs to be done on these to make them better. Now, the other thing to emerge from this field, and this is quite amusing, is that the studies have shown that if you, for example, you're treating inflammatory bowel disease and you give 100 people, FMT, 25% to 30% will improve, and the rest won't. And when they looked at the determinants of that, this is where the concept of the super donor has emerged. So there are some people sitting in the audience who are just champions. They produce some amazing poo that actually can heal things. And this is illustrated here by this. I chose this study because it's IBS, and that's relevant to the theme of the meeting. This was a study from Norway. And this is where they decided to adopt that concept of fixing the super donor. So this was a chap who was 36, very athletic. He was born the right way. He only had antibiotics 3 times in his life. Descendant of the Nordic gods. He was one of those kind of really, really healthy people, an athlete. And indeed, he donated poo for 18 months for the entire study. So this was quite an admirable job. And lo and behold, that was a positive study. So a lot of people actually improved when they received his poo. Now, that again is just telling us something. It's telling us that if you can match the donor with the disease, like that donor is replenishing something that is missing in that disease, that's very promising. And I think there's an opportunity for us to try and have a more intelligent approach to FMT instead of just giving it randomly from anyone and hoping for the best. I think this is a very important paper, very important concept. Now, the big question, though, is what is normal? I think this is really the crux of the matter, is how do we define a normal microbiome? So if we do analysis on the audience here, there'll be 100 different profiles. And looking at the mix of good guys and the bad guys is one way, but ultimately, it doesn't prove that you're actually healthy. It could be just chance. So for this reason, we actually, we wanted to answer that question in our center and design this study called the Healthy Optimal Australian Microbiome, the HOME study. And that's play on words because we want to bring people back home from whatever condition that they have. And for this, we started from the other end. We didn't start from which bugs are the best, which bugs are the healthiest, but who are the healthiest people in our society. So this whole study actually originated when I was giving a lecture like this. I think it was at the Garvan, and somebody asked, who would be your perfect donor? Like if you were to have an FMT, who would you choose as your donor? And my initial answer, well, I don't take shit from anyone, but if I were to do that, I guess it would have to be an Olympic athlete. Because Olympic athletes are incredibly fit, they eat well, they're lean, they're mentally very sharp and everything else. And then I thought about it more, and I thought maybe, they're okay when they're training, but when they're competing, they're very stressed. So I don't necessarily want to have, transfer that particular phenotype. And this is how this study was born. So we're really, recruiting adolescents all the way to 100 plus. The reason for that is that you could be very healthy at the age of 35, but you've not been tested by time. It's only those people that have actually survived to that end without dementia who are superbly fit. These are the fittest people in our society. So this study is looking at the entire spectrum, and hopefully it will provide us with the answers about what actually constitutes not just who's there, what bugs are there, but what they're producing, how they're transcribed, and so forth. So it'll be a very comprehensive way. This is the HOME study, so watch out for this space. Very important in this field that we nurture the next generation, because it's very important that we excite our young, talented researchers to go into this field, to learn the technology, to learn the bioinformatic analysis, all the difficult bits that people, old people like myself, brain is kind of completely saturated, but you have to invest in the next generation. This is why it's very, very important to excite our students, students even from school onwards. And in fact, we do have an example here, and this is one of the high school kids that approached us 4 years ago. And she wanted to learn about the microbiome, and she came and did a stint with us, Eunice, and she's also a top-class violinist. So this was her performance at the Sydney Opera House as a solo artist, which was tremendous. And she became a medical student actually with us at UNSW, 4th year, just finished her bachelor honors degree with us, and she'll be presenting tomorrow at this meeting. So I think this is really a great success story. I'm very proud of her. This is my last slide. This is just to show you our team. Always try and push the frontiers, but have fun at the same time and have a family. And this is our Microbiome Research Center. Thank you so much for your attention.
All right, thank you, Yemet, for that wonderful lecture. Now, I'm going to introduce our next speaker. And for those who had not had a chance to scan the QR code, on the top right, you see a QR code there. If you scan it, you can type in your questions in the chat group, and we'll have a panel discussion at the end of the lecture. Now, Professor Sunny Wong is from Singapore. Well, he works and lives there now, but he's originally from Hong Kong. Now, he's a very accomplished gastroenterologist, researcher. I listed the number of citations that he's had, the number of articles he's published, really to show that this is someone who, although that it's the same age as me, has already achieved what I could never in my lifetime. And he's also been given very prestigious international award. But what makes him so special? Now, despite all his achievement, he's the most down-to-earth, most friendly, humble person who's always eager to help. Now, a couple of years ago, when I had an opportunity to do a fellowship in Hong Kong, where Sunny used to work, and he was so grateful. I was so grateful of him that he sacrificed his own sleep, and he stayed in the hospital the whole night with me, doing emergency procedures on very unwell patients. And I think that there seems to be a recurrent thing here that because of me, Sunny has again lost a lot of sleep. He took a red-eye flight from Singapore last night and only just arrived this afternoon. So it really gives me great pleasure to introduce and welcome Sunny as our next speaker.
First of all, thank you, Peter, for the introduction, and also the organization for inviting me to give this talk. And it is my huge honor to be speaking in the middle of EMAT and also Nick. You remind me a lot about the photo because actually we were doing ERCP, and it was right in the morning. We were hungry, and we had a chocolate croissant and 2 cappuccino, which we just had half an hour ago. And I always wonder, like, how would these change your microbiome? So lots of sleep, traveling, diet. So all these are changing and are changing your microbiome. So I'm going to give the second talk, and the theme is on how to manipulate our microbiome. So thanks, EMAT has really set the stage on the background, introduction, and how important it is the microbiome. So do not need to convince you any further on this step, but we are going to on here look at how we can actually change the microbiome in a better way to make a healthier life. So a bit of a disclosure, I put up the same quote, I have 3, and so the first one is, of course, the most important by Hippocrates, that all diseases begins in the gut. And I do believe that, because we have seen that it is so important. So just a few slides to recap. We know the microbiome is everywhere. They are very important, but just remind you that it's not only in the gut. So although over 90% of them live in the gut, there are microbes all over our body as well. So they are in the skin, they are in the respiratory tract, they are in your nostril or your airway, and also everywhere in our body. So, but I would say that the most important parts of that live in the gut, so over 90%. And another thing that is important is because we know that although there are many trillions of them, the more important bit is that actually they have many more genes than our own. And because we know that these genes, they encode, they produce DNA, RNA, and the protein, and it is the protein that carry out some of the functions, the metabolic functions, or the other functions in our body. And they actually make 100 times more proteins than our own. So that's why we say that actually in terms of the function, they are many times than that we can actually produce by our own genome. So on the right, you can see some of these functions. So of course, in the GI tract, in the digestive tract, they are important in terms of the nutrition. We know that there are some vitamins that we cannot produce, vitamin K, for example, and we also know that there are carbohydrates that we cannot digest. So actually, we rely on these bacteria, these little buddies, to help us to convert these into nutrients that we need. We know that they are important. Emma has mentioned about fighting against infection, and it is because actually they being there, they actually train up our immune system, so they promote our immunity. And the other part of that is that there are some healthy little guys over there that actually they protect against the pathogens, the incoming infections. So it's also very important in terms of the immune. And of course, them being there, we know that they mature our gastrointestinal system. So for example, if we compare animals, what we call germ-free animals, so these are animals in the lab that were in the chamber, they were never exposed to bacteria in life. We know that if we look at the gastrointestinal tract, they are very different. So actually they are immature, and they have a very large cecum, for example. So they are they help to mature our intestinal system as well. So they are important in various aspects. Ima has mentioned about dysbiosis, and again, I agree with him that this is actually not the best word because this is quite vague, so we sometimes do not know what we are talking about. But I would give an analogy over here that the kind of dysbiosis, we commonly refer to these conditions. So when we say symbiosis, we refer to an ecology that is healthy, so balanced in a way. But if we are talking about dysbiosis, we can use to say that the ecology is changed. So it can be means to be a reduced diversity, meaning that they are not as diverse as they should be. And commonly we say about the balance as well. So there is expansion of the pathobiome, so they are the bad ones, or the loss of the beneficial probiotic or the good ones that we have. So there's a loss of the balance here that we commonly refer to. And the analogy that I would take is it is like a forest. So in a symbiotic, it is a healthy forest, like the one here. But if you have a fire, if you have disasters, so what you see is a reduction in the diversity or the richness. So in terms of number of species that you have is totally lost. And in terms of so what you are left is like a barren desert or a forest there. So I think this is the best analogy that you can have for dysbiosis. So how is it relevant? We know that it is associated now with every diseases. The most important, of course, are the GI tract because they are in the same organ. So in that, we talk about bowel cancer. We know that, for example, stomach cancer is importantly caused by bacteria called Helicobacter pylori. We also know that, for example, colon cancer, pancreatic cancer, there are some microbes that actually promote the cancer. So they are important in this manner. They are important in infection, like the C. diff infection that you have seen and I will talk about. Inflammatory bowel diseases because of the interaction with the immune environment. So actually they causes inflammation within the bowel itself, and also other like irritable bowel syndrome. So the gut is still a key organ, but increasingly we know that they are related to almost all other organs as well. The liver is related because the liver is part of the digestive tract, so it's part of our metabolic nutrition organ, so it is related. And we also know that there are connections between the gut and also the lung, the brain that Nick is going to talk about, and also the skin, and actually almost every other organ. And so you can see that, so there are, in each of these organs, there are diseases that are connected, autism, depression, stress, the lung, and also the other organs. So these are conditions that you will see. The other major bulk of the involvement is actually on the metabolic. It is because the gut is where we absorb our food, absorb the nutrients. So it is intuitive to say that microbiome is intrinsically related to obesity, diabetes, or these metabolic conditions. So the way that I see is, how would it be related to all other things, every other things? And the way I see it is that the gut microbiome is an organ by itself, and similar to the heart and the kidney, if there is something wrong with your heart or the kidney as a major organ, actually it connects to every part of the body, and this is how connected it is, and that's why it seems to be associated with every other conditions that you can think about. And so on this talk, we're going to have a look on what are major ways that you can change your microbiome, and I would think that actually there are quite a bit of different things over here. I would focus on 3 things because they are more important. Food and nutrition is important because that is the number one thing that you can actually do and change and also affect your microbiome. It is directly related because the microbiome are digesting the food that you're taking. Probiotic is one way that we know for a long time, and it is still a lot of research going on in probiotic as in taking in more beneficial species to tip the balance and make it more healthy. And lastly, on the FMT that we have seen and talk about as well, but this is getting so popular that you will see it on the news as well. Apart from that, there are other ways that you can see in this diagram. There are more fancy ways. For example, we can use antibiotics or newer antibiotics. We can engineer different bacteria to give you different purposes. There are phage therapies, so these are viruses that can attack some bacteria. So there are people also working on this very fancy scientific stuff, but I think they are not so ready. So I think for this one, I'll focus on the 3 I put on that I believe are the most important. So diet, probiotic, and also fecal transplant. So on diet, the first message that I want you to have is that when the moment that you're eating, your bodies are eating as well. So you are what you eat. So this is true, and so are the trillions of microbes inside your gut. And this is like hosting a dinner, so you are actually inviting the guests. And also the other message is that you got to choose what guests you are inviting. based on the food that you're taking, because there are different microbes, there are trillions of them, they like different food. So by deciding on what food that you take, actually you're inviting and selecting your own gut microbiome in a way. And we know that this is important because if you compare people in the rural area versus people in the urban area, they are very different. So for example, we know now enough that there are studies comparing these microbiome. So there were 2 major studies. So over 10 years now, you can see comparing Africans that live in US, in the urban places, and in really Africa, in the more native rural places. They show vast difference in the microbiome. So I wouldn't go into the names, but you can see apparently from the color that represent different microbes, the composition is very different. And on the right is another similar studies that compare urban places in Europe and also in rural area. And you can see from the color of the pie chart that they are very different between people that live in different places, in different geography, and taking very different diets. And I think that also come down to the kind of the chicken at the back. So you see actually on the plate is, you can see very much a plant-based diet, and if they were to take it from the chickens, it would be very fresh. Versus on the right, you can vaguely see M sign that represent a fast food shop there, and so you can imagine that the diets are very different, and therefore the microbes are very different as well. So you can see that the science has advanced over the past decade. So I'm showing that this elegant study that was in King's College in London. So what they did was they actually looked at over 1,000 individuals, and what they did was they profiled what they eat, they profiled the microbiome with the stool, and also they look at the blood as well. So this is a very complicated heat map, but what it shows you is that on the horizontal, you see different food groups. So meat, dessert, sugary food, and then you have the more healthy one, fruits, whole grain, nuts. And then on the vertical, you can see the names of different bacteria that are very complicated. So Roseburia, Clostridium, so all different names that you can see. But what you look for is actually the color of the chart. So actually what it tells you is that the heat map, the red ones are associated with each other. So immediately you can see that on the top right are the red ones. So actually these are bacteria that are associated with kind of the healthy ones that you have down there. So vegetable, nuts, fruits, whole grain, and so immediately you can see actually what it tells you is that if you eat unhealthy diet, as in the right and the left bottom, so you have kind of pathobiome, or what we say unhealthy bugs. But if you eat more healthily, fruits, vegetable, grains, you have more kind of healthy bacteria as well. So I think for the implication of this study is that I think it's good that in a way that for decades we have been told to eat healthy, and it does show you that eating a healthy lifestyle gives you a more healthy metabolic state as well. But what it adds is that it is also associated with healthy bacteria that you have. So it has been quite consistent that if you eat healthy, you have a better metabolic health, and also you have a more healthy microbiome as well. So just one more slide on the diet. So this is another quote from Hippocrates, and I think this is so true as well. Let thy food be the medicine, and thy medicine be the food. So actually, this is very much related, and food is so important that actually this is one, you can see it as one of the most important medicine that we have. But we have underscored the importance of it. So we have put the focuses on a lot of drugs, but we now know that actually what you eat is one of the most important things that you should do as well. There's one more slide on diet. So for example, you must have heard about something called Mediterranean diet. So this is a diet that is high in grains, fish, so kind of the typical, prototypical example of a healthy diet. So what it does is in this study, it shows you that if you eat, or if these people eat a healthy diet, so they do have a better outcome in terms of their cardiovascular risk. But what it shows you is that it changed the microbiome. But also, it tells you one thing is that actually the microbiome decides how much benefit that you're going to get as well. So what it does is, if they put them on this study for 8 weeks, so what they found is that, of course, it benefits the people. Depending on the bacteria, so for example, some people carry this bacteria called Prevotella copri, and what they realize is that if you are a carrier of these microbe, so you have a much more reduction in terms of the risk of myocardial infarction. So what you eat decides on your microbiome, but also the microbiome will decide how much benefit that you are going to gain from having this healthy diet as well. So there's a strong interlink and also relationship between them. So moving on, so I will go on to talk about probiotic as well. This is not something new, but I think there's quite a bit on the latest development that it might change the way that you understand the probiotic as well. So you can see that probiotic, if we look at it, it is live microbes that if we take it at the right amount, so you have a benefit. And now also we have a better term as well. So probiotic has been a term that has been used for decades. And so now we increasingly start to call it live biotherapeutics. And the difference is that in probiotics, we usually have a sense that it is over the market. It can be generally quite vague. We take it for general health. But in live biotherapeutics, we want to use these microbes for a definite indication or health benefit. So like a therapeutic that we use to treat a disease. So this is kind of the current concept that we have, and the field is moving into developing some of these beneficial bacteria or probiotic into biotherapeutics for a particular purpose. And I think the field is changing in a very rapid way. So on here is my final quote that is Prof. Michinov. So he was a Nobel laureate from Russia, but what he proposed at that time actually over 100 years ago was the concept of a healthy microbiome and probiotic. And he believed that it is possible to achieve a healthy life and also expanded longevity by modifying the gut microbes in our body. So there wasn't a term called microbiome at that time, but flora, basically the gut flora there, to replace it, to replace the harmful one by the useful one. So these are the probiotic that we have. So about probiotics, so one question, of course, is which one are we getting? So these are food that we know that are rich in probiotic, yogurt, kefir, and also kombucha, miso soup. So we have it in our diet already. And also we know that if we look at the probiotics, so these are some long names that you have. But these are the ones that actually we have been using. So they are not so new in a way, but they are many names, but I would sure that you probably would have heard about the lactobacillus, bifidobacterium. So these are the commonest type of probiotic we have, and they're off the shelf. They're just over the market. And we know that actually they are healthy, but what we have not been knowing is that actually they have been generally quite vague. So it improves your health, it improves your digestions, but we haven't been really using it for a strict indication or condition. So I would just give this example because although we still using these as probiotic, we are understanding a lot more from the research. So for example, these are all different kinds of lactobacillus that we all know about, and they have been there in the market for the past few decades. But I would say these are old dogs. So, but we understand quite a bit about the new biology. So taking example of colon cancer that we do. So for example, some of these lactobacillus, they were recently found to be really mechanistically, biologically very important in terms of the cancer as well. And that can be potential that they can prevent cancer. So for example, middle one is called Lactobacillus reuteri, and also on the right is Lactobacillus pentarium. We know that they are probiotic on the market, but what we understand actually both from this year and last year was that they were able to be in the gut, they produce some metabolite, and this metabolite can interact with the immune system, and these can actually prevent cancer in a biological way. Of course, we are not saying that immediately you can take this to prevent cancer, so I think there are still a lot more studies that we need to do in human, but at least in some of these laboratory studies in animals and other models, they actually show new ways, new biological mechanisms that potentially they can prevent the cancer. And apart from these old dogs, there are actually some new dogs too. So what are these? So there are what we call next generation probiotic. And on the right, you can see that there are some, again, very complicated names, but these are increasingly being known to be beneficial in our health, and they can potentially be served as a novel probiotic. So you can see some names, no history of use. One is called Akkermansia, one is called Bacteroides, and Faecalibacterium. So what are they? So actually, we know that they are probiotic, there are different microbes inside our body, but for a very long time, actually, we have been very hard to catch them. And the reason is that actually deep inside our gut, there are bacteria that are very oxygen sensitive. So imagine if you have some bacteria in your mouth, on your skin, they must be tolerating the oxygen because they are oxygen all around and they are able to survive. But actually, what happens is deep inside your gut, there are layers and there are areas that they are very oxygen depleted. So deep inside your gut, there are these bacteria, and we know that actually these are the important bacteria that helps to do fermentation and some other functions that you have. But because of the technology, we weren't able to find them and grow them, because if we take a sample, we culture them, there's environment, there's oxygen, so they will not grow. But what in the past a few decades was, we were able to take the stool, we do some sequencing, and find some of these very sensitive, oxygen-sensitive bacteria. We call them anaerobes, and we find that these anaerobes are extremely important to our health. So what technology now enables us to do is we're able to grow them in a chamber, so totally deprived of oxygen, and we know that actually some of these can have very drastic important functions in our health. So this is one that I would take an example on. So it was discovered not too long ago, only in 2004, but you can see that with the technology, we're able to understand a lot more about it. So even though it was only discovered in 2004, people have already completed the genome, so we know the genetic of this bacteria, we know a lot about the different functions, and we found that actually this bacteria is depleted in people who are obese, who have a lot of metabolic problems, and that's why. So if we, there are technologies, and there are also companies that try to make this into a probiotic for metabolic health. And it appears that there are some very good evidence. So looking at this bacteria, taking the oxygen and making them into a probiotic. So for example, this study published a few years back, looking at this Akkermansia in about over 30. So it is still a small trial, but it is the first clinical trial looking at these next generation probiotic. And so it appears that actually in only 30 subjects taking these probiotics, so these people, although they're obese or overweight to start with, they have improved insulin sensitivity, they have improved metabolic profile, and they have a better cholesterol control as well. So I think this field is still new. I mentioned that because you may see them, actually it is in the market already, so it's not widely publicized or known yet, but I think it is actually already in the market, and there will be more of these next generation probiotics. So this is something that you will see. We still need more data because this is only a small trial, but I think this is the first step forward. More names: Faecalibacterium pravanasi and also Roseberia. So these are promising ones that you may see, and in a few years you may see them coming after Akkermansia. But I think this would be actually a small step into the future. One challenge, of course, probiotic is not all rosy. One issue is that some people might take it differently. I have a very warm welcome from Peter today arriving here, but the question for you is how welcoming are your microbiome? So because when we take a probiotic, actually the microbiome, the already resident microbiome in your gut actually have to welcome them. So meaning that if you take a probiotic, if they are not welcoming in a sense, they will just reject the probiotic, and you will spend all the money, and then all the probiotic will be passed out in the stool. So that doesn't serve any purpose. So the key is we need to understand a bit better. So in what people, so we know that this is highly personalized. So when we take probiotic out of 100 people, not all of them will stay in the gut. So I think this is a major challenge for the field of probiotic that we need to tackle. So I think lastly, just on fecal transplant, a few slides. So this is something that is important and also is on the news media because this is very eye-catching in a way. And I still remember when we first did the FMT a few years back, we actually bought a blender, so it's a household one, and also got a coffee filter paper. So it's really gross, but the field has also advanced that we have a much more better technology to keep the stool, to freeze it, to make it into capsule that makes it more elegant in a way. Ima has mentioned that we use it for C. diff infection. It is miraculously effective for this condition. So this is the paper that initiated all these FMT studies. You can see that it is 2013, exactly 10 years ago, and the field has moved so much forward now. So we know that the cure rate at that time is about 90% versus conventional rate of 30%. So this is how miraculous it is for C. diff infection. But we also know, and so that because of these miraculous improvement, we try to make it into and applied it for other conditions. And you can see that metabolic, obesity, heart diseases, we try to apply that. But as Emad has mentioned, it is not at all very easy for these conditions because these are all complex conditions rather than a simple C. diff infection. So these are some of the trials that you can see that people have tried and studied on. So apart from C. diff, ulcerative colitis, Crohn's disease, these are inflammatory bowel diseases. There are other bowel diseases, there are metabolic diseases, and there are people even trying in autism, for example. But one simple message is that they are not as easy as C. diff. You can see on the right in the small print that they are harder, the clinical efficacy, the benefit is not as clear. And so for example, take one example is metabolic syndrome. So people have done an FMT trial in these people. They show better in terms of the insulin sensitivity, but on some of these studies, there were no significant change in the major phenotypes. So for example, the BMI, the body mass that they have. So one message here is that it's not all ready for all the other indications that you see. And there were also a bit of safety reports as well. Could have, generally they are quite safe, but you could have infective and other complications from that. So the simple message is don't try it at home by yourself. And, but I think the field is moving forward. There are better alternatives. And you may see on the news as well that in US, they approved recently 2 microbiome-related therapeutics. So they are more advanced version of FMT. Some of them take a consortium of bacteria out from the stool, and then they make it more elegant. So these are on the news as well. So I think the field is still moving forward positively, and there are much more research that's going to happen. So I will summarize that microbiome is important. It can be modulated, and I think the best way is actually the way you eat, the gut diet, and also other ways are probiotic FMT. They do confer a health benefit, and there are also potential for next generation probiotic. And regarding FMT, I think we do want to know more about it, apart from C. diff as well. And so this is my institution in Singapore. So if anyone happens to drop by, so do come for a visit. Thank you very much.
Okay, thank you, Sandy, for that lecture. As you were talking, I was wondering if anyone had actually seen how a fecal transplant is actually done. Well, I have, and currently the way that we do that still involves big syringes of poo, and we have to thaw them because they're completely frozen. We have to thaw them for a couple of hours, and we do a colonoscopy. Those are putting a long chip of video camera into someone's colon, and then we have release the poo into someone's bowel. So it's quite an interesting process, but I think I agree with Sunny that there could be a more elegant way to achieve the same goal. I want to now introduce our final speaker of the evening, Professor Nick Talley. I already had his permission not to list all his achievements because we'll be otherwise here for the whole night. But this guy has his own Wikipedia page. And you know that means serious character. So I encourage you all to go and read up about his achievement thus far. I will say one thing though. He authored a very important textbook that every single doctor in this theater had read. And I had also held on to my copy for almost 25 years. And even though I have moved houses quite a few times, this is one of the very few textbooks that I kept referring back to, and I just could never part with it. And last year, this year, beginning of this year, I finally was able to get Nick to autograph my textbook. It's now a very prized collection of mine. So please join me to welcome Professor Talley. Run? Do I? Okay.
So thank you, and good evening, ladies and gentlemen. So I was racking my brains what to talk about after the last 2 speakers have said everything about the microbiome and done it very, very well. So what I've decided to do is to talk about some of the research that my group has been doing to try and work out which of those microbes might be the bad guys. And if we can work that out, potentially we might be able to cure disease, not just treat things, improve things, but really make a difference. Some of the work I'm going to present is new. It's been presented at major international meetings. but some of it's unpublished, so you need to know that. But there's lots of it published as well. So I'll take you through a bit of a story. I hope it'll be of some interest. And for those in the audience, the disorder I'm talking about is very common, so we'll cover that as well. I want to pay my respects to the traditional custodians of the lands on which we meet today, and pay my respects to elders past and present as well. I have my disclosures here, including a few patents, which you can read at your leisure. But let's talk about a condition that is really common in Australia. Lots of people have this, and really it's a condition that I've spent a long time thinking about and talking about. And these are unexplained upper gut symptoms, or what's often referred to in the medical literature as functional dyspepsia. Now, Jennifer Hawkins, as far as I know, does not have functional dyspepsia. I want to be very, very clear about this, although I have a little bit of a story to tell about why I've included her photo. But this is a very common syndrome. So look, in this audience, I estimate that there's probably about 10 or 15 of you with the condition, and another 10 to 15 are going to get it. Isn't that exciting? Isn't that a thrill? And this is a very unpleasant condition. This is a condition that causes fullness and bloating after you eat, so you can't even finish a normal meal sometimes. It helps you to lose weight, but that doesn't make you happy, I can assure you. You may have stomach pain and stomach burning. You may have nausea, you may have some bowel disturbances as well, because this condition overlaps with the irritable bowel syndrome that probably most of you at least have heard about in your daily life. This condition affects quality of life and work. You often have headaches, backaches, fatigue. You feel unwell, lots of systemic symptoms, anxiety and depression. Anxiety in particular is very common. It's about 1 in 6 Australians if you look at the epidemiology data. And of course, we don't know the cause. That's why it's called unexplained. If you don't know the cause, you can't cure it. We know the microbiome's linked, so we're going to talk about that. So why Jennifer Hawkins? Well, I guess I'm often asked why I became interested in this condition. And that's because when I was a resident and then a junior registrar, I had a girlfriend and later fiancée called Jenny, and she actually had this syndrome. I've never forgotten it. It was absolutely awful. Seriously, it was really, really awful. Now, I've got a very sad end to this tale because my Jenny actually got killed in a motor vehicle accident, so it's very sad for me. But I decided at that point I was going to do something about this, or at least try to. So that's why I've included that photo. So I'm going to show you the best paper on functional dyspepsia that's been written in recent times. It's a very good paper. I love this paper in the British Medical Journal, one of the best journals in the world, the BMJ, and here it is, published in 1910. Functional dyspepsia in 1910. You know how they diagnosed it? They opened you up to make sure you didn't have an ulcer or a cancer. Can you imagine? You had an operation to work out if you had functional dyspepsia. And of course, lots of people did. And this article describes, the condition, the symptoms, and some of the potential, what they thought might be the causes, which I'll come back to. So this is a common condition. This affects lots of people. We understand there are subgroups of this condition, and we're starting to understand a little bit more about it because we've done a number of studies. And this is my favourite study that I ever did, ever. And this was a study where I went to Sweden. It was a terrible ordeal, really, having to go to Sweden. Went to Sweden, and we invited the participants in a town, 2 towns actually, to come into a research study, randomly selected. So basically, at random, we invited people to come in to this study. And we sent them a nice letter, and we called them up, and we had ethics approval and all the usual things. And we spoke to people, and people seemed really interested. So we couldn't believe it, because what we were offering them was a free T-shirt and a free endoscopy upper endoscopy. Down this end, unsedated. Yep. It had to be unsedated. That was the ethics committee. It was required. Unsedated. So we approached all these people at random. And of the people we approached, 80% said, yes, we'll come in and have our free scope. Yes, please, let's do it. And they came in, they had the free scope, we biopsied everything, we took samples, we took blood, all the usual things you do. And we showed 1 in 6 have functional dyspepsia, just like I talked about. And this is really one of the gold standard studies that tells you about this condition. It's a very, very common condition. And what are the causes? Well, the 1910 article talked about a lot of the causes that we've all talked about until about 15 years ago. Talked about anxiety, talked about inflammation in the stomach, gastritis, talked about acid. You heard about stomach acid, how important that is. Talked about motor disturbances, the stomach not working properly. Even talked about the microbiome, fermentation in 1910. Can you believe it? Nothing changes, ladies and gentlemen. We just go around in circles and we do it very well. We just give different names to it, of course, because that makes it more interesting, but we don't always progress everything. So really, and look at the list here. This is the list we had, recently. Very, very similar. Gastric acid, Helicobacter pylori, which is a cause of gastritis, of course, and peptic ulcer and gastric cancer, but it can also, at least some people believe, cause symptoms without those things, and some of the other motor mechanisms and sensory mechanisms. So, you could stop here and say that's what we knew about this condition, until very, very recently. And essentially, when you look at those mechanisms, we can't fix any of them. We can't reverse almost any of them except Helicobacter pylori. That's the one we can treat. It's a bug, so we can treat it. So what happens if you treat Helicobacter pylori infected people with functional dyspepsia, which is, by the way, a minority of cases? So what do you get? And the answer is you get a benefit. You do. This is a forest plot for those who want the technical term, looking at multiple randomised controlled trials. But what's really interesting is there's no correlation at all, essentially, between eradicating the bug, that means getting rid of it, curing it, and the symptoms improving. Doesn't correlate. They get better, but is it because of the Helicobacter pylori? It's very interesting. And some people have suggested, well, Helicobacter pylori is very important, but there's other evidence all over the place that Helicobacter pylori isn't associated with dyspepsia symptoms when you look at it in clinical studies. But if you give antibiotics, some of these people do get better. But maybe it's not the Helicobacter pylori you're treating. Think about all those other microbes in the intestinal tract. You've heard about all those trillions they've talked about, my colleagues, trillions. Maybe there's something else being hit. We like that hypothesis. We really like that hypothesis. There's some other really interesting things. You heard about this as well. Asthma's linked to functional dyspepsia. Isn't that interesting? Asthma. Why would that be? Why would asthma be linked to this? And we know asthma is an allergic condition in some people, an inflammatory condition in some people. It's actually a condition in some people linked to some white cells that are increased in the lung called eosinophils. So it's interesting that's the case. And functional dyspepsia is increasing, which is all, at least in Sweden, which we have wondered about why that might be the case as well. But by the way, SARS-CoV-2, that's COVID-19, it's a risk factor for getting functional dyspepsia and irritable bowel. Isn't that a cheery thought? Isn't that great? All of us have had this infection and it will lead in some people to these gut problems. Oh, it's so exciting. Oh, it's good for me. I'm going to be very busy for a long time, and so are my colleagues. Okay. So we, in that study I talked about in Sweden, the one where we got the random sample of the population, we decided we'd look at the stomach and the intestine, the upper part of the small intestine, what's called the duodenum, we would go there and we would look for evidence of an allergic inflammation, a bit like that asthma business I talked about. And that's exactly what we at least found in this study. We found this increase in these white cells called eosinophils, which really hadn't been appreciated, at least by very many people, prior to this particular study. And we also showed that these eosinophils were active and they led to the release of toxic proteins that could potentially damage nerves. And we've shown very similar results here in Australia in a number of studies. Here's one of our most recent studies showing again this increase in these white cells in this upper part of the small intestine, the duodenum, and also a leaky gut. The duodenum is leaky, just like you heard about already, as an important mechanism in disease from EMAD. So yeah, we think that's right. And others have also shown that there's damage to nerves in this part of the small intestine. This is work from Belgium, a very distinguished group of researchers led by Jan Tak. And basically they also showed the nerves not only were structurally abnormal, linked to the inflammation, but they were functionally abnormal. Yeah, so very, very, very interesting results. We've also shown in the blood there's evidence of this inflammation in the blood. Now remember, functional dyspepsia is called functional because it's not supposed to have any pathology. It's not supposed to be inflammatory. It's supposed to be something else. So that's what's so interesting about this. The complete term functional just doesn't seem to fit. But there's good evidence of these small intestinal homing T cells, and that'll become important in a few minutes when I talk about bugs and this disease. So just remember that. What's interesting too about these small bowel homing T cells is it correlates, it's linked to, we think, small or gastric stomach emptying. So stomach emptying is slow in some of these people with functional dyspepsia, and the more inflammation you have, the slower your stomach empties. So the duodenum, the upper part of the small intestine, is affecting not just that part of the gut, but also the stomach as well, potentially, which I think is very, very important. We can show similar results in animal models. This is a stress model, interestingly, where maternal separation of rats. But what's interesting about this model is if you do that, you can show in these rats, they get these white cells increasing in the stomach and duodenum. And this is also linked to changes in gut function. So it's not just in humans, even animals, this can happen. And stress could be one of the factors that drives the condition. And then we did this really interesting study in the United States most recently, where we actually went to gastroenterology practices, 20 across the country, and we biopsied their stomach and their duodenum, their upper small intestine, looking for these inflammatory cells. And we were particularly interested to see whether we would see these linked to these unexplained gut disorders. So we recruited patients and controls, and the patients had unexplained gut disorders: reflux disease, irritable bowel syndrome, functional dyspepsia. And what we found is 40% of those patients, 40%, had these inflammatory changes in this upper part of the small intestine. Okay, so, and then we also looked for mast cells. For those who are interested in mast cells, they were slightly increased, not very much compared to controls, interestingly. They were up, but not very much, but they were activated when you look at them. So the mast cells were ready to explode, as were the eosinophils. This was tested by flow cytometry in this particular case. So what's been interesting about this is looking at the association of this inflammation in the upper small intestine and other manifestations that these patients with this condition can present with. For example, reflux symptoms, heartburn. And what's interesting is if you've got this inflammation, your six-fold increased risk, or 600 times increased risk of actually developing reflux within 10 years, even though you had no reflux at the time you had the biopsies taken. And similar results for anxiety. In other words, if you had this inflammation, you were more likely to develop anxiety. So now we've got a targeted area of the gut to focus on. That's the whole reason to do this work. So what we want to know now is, why is there inflammation? And could it be that this part of the gut is the central part, the centerpiece of why you develop this unexplained set of gut? And actually, not just gut, other symptoms too. And so we're very interested in looking at what might drive this. And I'm going to focus on the microbiome and food, because everybody's interested in those things, and that's what the relevance of today is. So what we did was, first of all, this is Gerald Holtman's invention that I was involved with, but really Gerald drove this. And this was to develop an aseptic biopsy device so we could get sampling done that would actually be cleaner than just using a standard biopsy forceps. Because when you put this thing through the scope, it gets dirty as you go in and out, or at least it could. So that was one advance for the group. And then we decided we would look at the duodenal, the upper small intestine, microbiome. We would target there because that's where we found the inflammation, and that was therefore the most sensible part to go. And what we did is we actually took biopsies and we looked at what's called 16S, but regardless of the technique, we looked to see what was the relative abundance of different bacteria. So we were really interested in seeing whether we could identify a group of bacteria that might be linked to the disease of interest. And frankly, it hadn't really been looked at before. And what we found, very interestingly, and what we've confirmed in a subsequent work just published again, is an increase in streptococci, what I thought to be oral streptococci, in the actual duodenum, the upper small intestine, and actually suppression of some of the other bacteria compared with healthy controls. And the more bacteria overall you had in this area of the gut, the more symptoms you had and the worse your quality of life. Forget about that. Too technical. We next looked at whether there was any link to dietary factors, and we focused on those who said they were wheat sensitive. And interestingly, the group who said they were wheat sensitive had the dominance of these streptococci. Again, there were other things too we could have focused on, but the streptococci stood out, at least in our minds, as we looked at this, because we know from other work that streptococci can break down gliadin and release some of those antigens that potentially could interact with the immune system of the gut. Yeah. So we decided we would try to culture the streptococci. Remember, we've got an area of a target, we've got now a group of bugs we're interested in, but we haven't actually grown them, we've just shown they're there. We wanted to grow them to see if there's any novel bacteria that are present. And this work's been presented at Digestive Diseases Week and UEGW and a few other places, and it's been led by Mark Morrison and Emily Hout and some other key people. Emily's with us in Newcastle. Bottom line is, we grew 3 completely novel streptococci. Completely novel. Yep. And we DNA sequenced them. We sequenced them to see what they were. And what was really interesting about these are they are different, and one of them in particular had a virulence factor that looked quite interesting, at least to us. And this is how we actually did this, and I'm not going to take you through it because I don't think this is the right place to do so, but for those who'd like to see what we did, this is what we've done. And basically, it allowed us to identify these bacteria. This is what's called culturomics. What excites me about this is we're looking for bugs that might cause disease, not looking at the whole of the microbiome, which you've heard my colleagues talk about so elegantly, but looking for a particular bug that causes a particular disease that nobody's found before. At least I'm excited about that, even if you guys aren't. I'm excited because if you find a bug, you can treat it. Maybe. Okay, we found this bug induced an immune response. Here's some detail, I won't take you through it. We found some of the proteins that were inducing what was the seroreactivity, the immune response that we're able to measure in the blood, which I thought was exciting. And this is just more evidence of what we did. So again, the detail's not relevant. And then Jasmine Walk, who was doing her honours with us and is now a medical student at Newcastle, she identified that this one of these particular novel streps actually had a unique gene, virulence gene, could translocate, could punch holes in cells, in cell layers, and could actually reduce duodenal barrier function, reduce the proteins there that protect the barrier, which we thought was very exciting, and this is what she presented at UEGW just a few weeks ago. And bottom line is, we think this is a very good candidate bug for driving disease. And what's very interesting about this bug is it's sensitive to amoxicillin. And we use amoxicillin with other things to treat Helicobacter pylori. And we're coming back to the story that sounds, at least to me, convincing. And our next step will be that randomised trial to find out if that's really true. And that will be our next step. So I mentioned foods, and I'm going to be very quick, but it's been really interesting. It's been a revolution in the thinking about food and gut disease. Because in these unexplained gut syndromes, now we know from work, elegant work from others, work that many of us are now working on too, we know that if you put the foods into the upper small intestine, you get an immune reaction in some people with these conditions very, very promptly. And it's characterised by these white cells called eosinophils in part. And what's interesting is we normally scope people fasting. And if you take the biopsies then, you don't see much. You need to give them something that stimulates their system to drive the pathology that we identified, luckily, from the previous studies. Quite honestly, it was luck, but I like luck. Luck is good in science. So this is really interesting work, and wheat is one of the keys, but it's interesting, these are really being seen in functional dyspepsia and irritable bowel syndrome now. Not everybody, by the way, about half of the patients. So not everybody has this mechanism, but about half of them do, at least according to the work. And what's interesting is the foods that do this are the same foods that cause what's called eosinophilic esophagitis, which is an eosinophil disease of the swallowing tube that causes trouble swallowing and is now getting more common. So this is a fascinating piece of sort of new work that links in, we think, to the microbiome as well. I can't take you through all the rest of it, but this is the working hypothesis that you actually get digested gluten peptides because of the bugs. We think streptococci could be a key one, but we have to prove that. And that then leads to an immune response that sets off what is a systemic disease called functional dyspepsia, which isn't functional at all. And what's interested us as well is there's some other work suggesting some of the dietary factors and other cells that infiltrate, as well as the bugs, can all produce histamine through various mechanisms, and blocking histamine seems to be actually an effective sort of band-aid treatment, but very cheap and easy to do. And so we've got some data suggesting that may work in functional dyspepsia, and there's been some lovely work in irritable bowel syndrome as well. So I think that's going to be a new area. So what I've said really is we've got an unknown disorder. We've identified risk factors from research done over decades, And now we're starting to understand that all of those risk factors actually can be brought together into a working hypothesis that ties everything together, explains everything, the symptoms, the whole of the sort of pathology, the changes we see objectively. And I think that's a very exciting thing. And this is sort of what we've put into the literature that's been published in a few places, very similar diagrams in these places. And bottom line is we think the inflammatory process, of course, is important, but the drivers of that inflammation, that's where we can really achieve potential for reversal of disease if this working hypothesis is right. So what I've tried to tell you is it's possible, despite the trillions of bugs, to actually work out, potentially anyway, theoretically anyway, what might be cause and effect. Now that's very different to a lot of the microbiome research that's done, looking at multiple bugs changing up and down in the microbiome, links to disease. But the problem with that work is it's all association. And for the scientists in the room, you know association doesn't necessarily mean cause. Now, I also want to caveat this. Some of this is hypothesis only at the moment. It needs to be proven, and it will be, either one way or the other. It'll be positively proven or it'll be disproven, and that's what you need to do. But I still wanted to emphasise that at least I hope it illustrates an approach to the scientific method you can apply that's lots of fun and very, very interesting. So I often am asked, could eosinophils be an explanation for some of these unexplained gut disorders? And I loved a journal reviewer who wrote this in a reply once to one of the papers we submitted, where he or she said a disease which is considered rare isn't looked for, a disease that isn't looked for isn't diagnosed, and if you don't diagnose it, it must be rare or doesn't exist at all. And I think that's really good. So a few take-home points, which I've already kind of covered, but I like the guy on Twitter who said he found out he has FD, functional dyspepsia, but he's got a mechanism. And even that made him feel better, even though it didn't cure him at all. So I like that a lot. And let me say finally, thanks to the group I work with, who do all the work, are just fantastic, and my collaborators and colleagues and friends. Thank you very much indeed.
All right, now that we have heard from all three speakers, it's time for our Q&A sessions. Look, what I'll do is I'll alternate between the live questions and the sort of entered questions on the website. So does anyone from the audience wish to ask questions? Yes.
Do you think microbiomes can one day help detect or diagnose cancer?
Thanks so much. Excellent question. Absolutely. Most cancers, certainly over the last 15 years or so, have a very unique microbiota associated with them. And in some cases, actually, it's initiated by an infection. The classic one is stomach cancer, which is initiated by Helicobacter pylori, a very common infection that half the world has got. But when it's present in the stomach, it causes chronic inflammation. Remember what I said about the root of all evil is chronic inflammation. And then that progresses, and then it accumulates, all the changes that culminate in cancer. The same applies to bowel cancer. The same applies to many other cancers in the human body. It is possible now to actually have a very unique fingerprint based on the microbiome that can predict a specific cancer. And this work is very exciting. It's been published in top-class journals, and it would be possible to use it not just for diagnosis, but also for prognostication. You know, how you're going to respond to treatment, for example, whether it's immunotherapy or chemotherapy or radiotherapy, for prediction of side effects of that treatment, and also for screening in the population. And in fact, Sunny, sitting next to me, has been one of the world's leaders in relation to that particular aspect in relation to colon cancer. Perhaps, Sunny, you want to add a little bit about the colon cancer work that you've done?
Yeah, thank you. So we did publish some work. So I think the idea of a biomarker is that when cancer develops, so there are different components. So there's the genetic, so there's some diet, and microbiome is obviously one factor. And so the idea is that actually if the microbiome is an important factor, so so the cancer during its development, it will leave some traces. And if we find these traces, then we were able to help us actually develop new biomarker for cancer. So for example, what we did was we compare people with and without cancer, and we found that some of the bacterium, including one called fusobacterium, is increased in people with cancer. So if we take that forward, if we take the stool, if we look at these fusobacterium, we're going to see an increased amount of these fusobacterium in people with the cancer. So that's why we can actually detect them, and that would help us diagnose the cancer. So I wouldn't say that it will help 100%. So everything in life is, especially with biology, they are multifactorial, but it definitely would add to what is existing. So what we showed, for example, is that if we add this marker, fusobacterium, to the occult blood test that we have, so you would increase the percentage that we'll be able to screen for the cancer. So these are some of the ideas that we can develop and work further on.
Okay, thank you. Now, I'll follow this up with another question. This is an online question along the theme of Helicobacter. Now, you have mentioned that Helicobacter causes cancers, stomach cancers, possibly bowel cancer as well. The current treatment is antibiotics, which also sounded like something that's not going to be very good for your gut. So, is it worth treating the Helicobacter with the antibiotics?
Maybe I'll start first. I think so, because we know that actually Helicobacter is one of the most important causes of stomach cancer. And in fact, if we look across all stomach cancer, so it's about 90% is caused by Helicobacter, and it's classified as by the WHO as a class 1 carcinogen in a way. So that's why if we diagnose it, even if a patient without cancer, we tend to eradicate this, and they will reduce the further risk of cancer. However, we know that this is not the complete story, and we know increasingly that Helicobacter pylori is an important part of the story, but it's not the whole story. So we know that people who have what we call intestinal metaplasia, so they are halfway, so they're actually what we call pre-malignant condition. It's not cancer, but the person have already in the stomach, there are some changes that, so these people have increased risk of cancer. So even in these people, if we eradicate the bacteria, it doesn't prevent or totally negate the risk of cancer. So we believe that there are something else in the stomach. So there may be other bacteria, there may be other genetic or other not genetic factor that's important as well. So simple word is, I think that should be eradicated if it's a case of Helicobacter pylori, but this is not 100% of the whole story. And I just wonder whether you had something.
Yeah, so just two aspects.
Up on the screen.
Two aspects is, one is the Helicobacter progression. So half the world has got it. Obviously, a tiny proportion will end up developing gastric cancer. So the majority of us will have no negative outcome at all. However, the way that it works in some people is that it causes that inflammation, and in time, you start losing your capacity to produce acid. Remember I said that it's very, so if you lose that first line of defense, again, you're not going to have any symptoms at all, but what's growing in your stomach is a very filthy microbiota that sustains that inflammation. So even H. pylori disappears in those latter stages, but you've got all this kind of mix of other microbiota that can drive it. Now, I think Pete's question was about treatment with antibiotics. Is that actually worth, antibiotics obviously can affect the microbiome. If you're trying to prevent cancer on a population-wide, are there any negative effects? Most of the studies that have been done so far suggest that, in fact, all the negative effects of eradication have not panned out in population studies. For example, there isn't an increased risk of reflux or developing esophageal cancer or developing negative effects on microbial resistance. All of these things, and when you give antibiotics, you're worried that it might increase the... so these studies have been done, and we collaborate with our colleagues in Taiwan who have been doing these studies for a number of years, following eradication therapy, looking for antimicrobial resistance, looking for any changes in the microbiome. All of these changes are very transient, and then once 8 weeks to a couple of months, everything goes back to normal. So there's no concern in relation to the negative effects of antibiotics on the microbiome. I'm quite happy to get rid of H. pylori to prevent stomach cancer, particularly in populations that have a very high incidence of stomach cancer. China, Japan, Korea, this is one of the biggest killers. And one of the biggest advances in our specialty in gastroenterology has been the reduction in the incidence of gastric cancer by eradication of H. pylori.
Okay, thank you. Thank you. Now, yes, please.
Okay, thank you. This is a few questions for Dr. LMR, and two small questions and a big one. The two small questions are relatively personal, and if you wouldn't mind, and they are you a vegetarian, and have you always been a vegetarian?
Am I a vegetarian?
Yes.
No, I'm not a vegetarian.
Okay, that's okay.
I eat healthy diet, but my diet is actually very, very simple. I eat what I want to eat and avoid the obvious,
I see.
excessive red meat and so forth. But I do actually consciously increase the number of items that are plant-based. So I do follow that very, very, and in fact, my breakfast, which is made of, I live most of my life in Scotland, so porridge is the basis of it. Adding nuts and fruits and vegetables, there's about 15 out of the 30 just in that bowl that I consume every day. So to some extent, I think if you follow that, you could probably overcome the contribution of some, everybody likes a steak, once in a while, and other things that might, like chocolates and the things that you might have tomorrow at dinner. But that's fine, as long as you have enough healthy ingredients that can feed your microbiome. Actually, I'm quite satisfied with that.
Right, I see. That's astounding. And my third question might have been partially answered by what you've just said. It's a little bit of a playing of a devil's advocate, because you mentioned how meat can partially contribute to gut inflammation and, in a way, contribute to IBS. And I just wonder, is there a possibility that, say, Mr. Evolution have prepared us the coping mechanism for the inflammation that is induced with, let's say, meat-heavy diet, and once we actually remove that, either completely eliminated or reduce the intake of meat, that somehow make us weaker in terms of our coping mechanism?
That's a very intelligent question, actually. In other words, if you look at our evolution and throughout history, so we were hunter-gatherers, so we're eating meat and berries, by the way. So that combination was always probably ideal for dealing with the challenges then. Yes, I take your point about. That's why I don't advocate a particular diet, like one specific diet where you exclude certain things completely, because there's probably some benefit from eating red meat. There's no doubt about it. But if you're eating red meat every day, you will be producing metabolites that are not particularly healthy. And if you look at population studies, you'll see that the higher intake of red meat correlates with certain diseases. That doesn't mean that on an individual basis that it's not giving you some benefit of some kind. It probably is protecting you against something that we don't know. So from that point of view, I think that's why I believe in the concept of that mixed diet. It's not, you're not vegan or you're not vegetarian, you're excluding everything. Just common sense, and enrich it in the ingredients that we know will actually give you a much healthier, balanced microbiome.
Thank you very much.
Okay, thank you. Our next question goes to Professor Talley. It's an online question, and I'm sorry if I paraphrase your question incorrectly. Nick, what is can you get functional dyspepsia while on a gluten-free diet? What is the role of gluten-free diet in this whole functional dyspepsia? Is it through inflammations or modulation of microbes?
So it's a very good question. Lots of people think they are gluten intolerant who do not have celiac disease. That's very common. And lots of those people also report unexplained gut symptoms, which sometimes improve when they take wheat products out. But it's a very complicated story. It really, really is hard, because there have been studies done where they've blindly taken out wheat products from people who thought they were wheat sensitive. And some of them were wheat sensitive, about 20%. But the others actually really weren't. They did not seem to be genuinely wheat sensitive when it was blindly reintroduced. So that makes it really hard to work out what's going on exactly, and it's very much an individual issue. So yes, you can still get unexplained gut problems even if you take all the wheat out. A gluten-free diet is not a healthier diet. I'm really sorry to tell you, unless you have celiac disease, in which case it's a much healthier diet. But if you don't have celiac disease and you're on a gluten-free diet, it's actually less healthy. You've got more cardiovascular risk on a gluten-free diet if you're not celiac, for example. So we want to be right about taking things away that really are important to take away, which is why I guess we're doing some work to try and sort that out. But wheat protein certainly can induce an inflammation in the gut or an immune activation in the gut in some people that may be relevant.
Thank you. Any other questions from the floor?
I heard. I think. Can you hear me now? Oh, perfect. For Dr. Telly, first of all, thank you for coming here. With regards to functional dyspepsia, I was very interested in the focus on streptococcus. I just wanted to ask, how do we know that that's not the effect in terms of the people that would test it, in terms of like habits and their lifestyle choices and how much stress they take? How do we know that that doesn't factor and influence into them producing that bacteria more than other people?
Thank you. It's a very good question. It's the chicken and egg question. And it's a very important question. And what you need to do is fulfill Koch's postulates, basically, to test if a bacteria, for example, is the cause of the problem or secondary to something else which goes along with the problem, which is really what you're asking. So we've fulfilled some of those, but we haven't done the most crucial, and that is get rid of the bacteria to see what happens. And that's the most important next step. But you're quite right. It could be you see more of these streptococci, even though they're novel and even though they induce an immune activation and even though they seem to have antibodies and all those things, that could all be because of some other factor linked to the disease and the presence of the bug. For example, some dietary change that's relevant. We don't think that's likely, but it's possible, we agree.
Thank you. Next, anyone else want to ask questions from the floor? Michael.
In transplants, you use patients use anti-rejection drugs. Can microbiome be used instead?
And that, again, that's a very intelligent question. I think you're all asking the most important questions. There is no doubt that the microbiome will impact on your immune system and your response to. So transplant, obviously, you give something to suppress your immunity to allow you to accept the graft or the organ. Whether you can complement your immune system or skew it in the right direction so that it becomes receptive is actually a very intelligent suggestion, and it's something that I know that there are certain groups who work with transplant are actually working on this. So it might be a kind of an adjuvant treatment, something that you can give at the same time as a transplant. There is a lot of interest in actually predicting success of transplants based on your microbiome at the time of the transplant. So it's very conceivable that it might be manipulating in the right direction if you understand which way you need to skew it to allow the success of the transplant, that, and so forth. So it's a very valid question, very valid pathway for research, and it requires a lot of research to be done.
And there's a second question. For those fecal capsules, can they be used to cure specific disease like kidney CKD or something like that?
So that's the kind of the ambition, is to be able to, by manipulating the microbiome in specific conditions, not just C. diff. We talked about C. diff, it's very easy, you're just overwhelming one particular bug. Inflammatory bowel disease is one that has benefited a lot from this particular approach. Obesity, dementia, autism, all of these things. There are more than 350 clinical trials on FMT in so many different conditions. The problem that I have with most of these trials is that it's still, the approach is kind of random. You're hoping that this is actually the optimal donor or the optimal transplant. And I think we need to be much more intelligent in our approach by understanding the signature of the disease, what's missing, and what needs to be replenished, and then doing like a cross match. It's like, when you do a transplant, you match. You're not going to get a perfect match for you so that this engraftment is actually successful. So it is, this is the aim of most of the research in this field in FMT, is to try and expand the indications for transplant and success of those transplants.
Okay, we'll do one online question. Now, the professors have promoted the plant-based diet, and why? What is it about the vegetables and fruit that are good for us? Is it the fibers, is it the nutrient, or is it the microbiomes of the plants that we're eating? The second part of the question is, does it make a difference if we're eating organic products?
Yeah, I think what you mentioned are correct. So there are a few components. We are talking about the food. So the food has the nutrients that we need. We're talking about the food that could affect the microbiome. So microbiome can have a big role. And we are also talking about the microbiome metabolites. So these are after the microbiome has digested the food. So what do they give? And we know that from various studies that all these three components are important. So for example, if we're talking about the plant, we know now enough that there are microbes that can ferment some of these fiber. And we also know that different plants, they have different fiber. And microbiome can metabolize them in a different way to give different metabolites. So like what Emad has mentioned, short-chain fatty acid is a large group. So that these are metabolites that can only be produced by microbes, but not ourselves. So after fermentation, so they can actually digest this fiber. They give these short-chain fatty acid. These are tiny molecules that are small, between carbon of 2 to 4 carbons. So examples are butyrate, and we know that examples that butyrate can have anti-inflammatory, it can slow down some cancer, and it has a beneficial body function. So I think all you mentioned are correct. So the fiber or the nutrients are important, the microbes, and also some of the metabolites that produce. Again, I guess with respect to what you asked about the fiber, so these are the examples of how it is important. And conversely, if you're talking about red meat, we also know that microbes can digest them. They can give some metabolites that has been shown to be harmful. They can promote atherosclerosis. They can promote platelet activation that are important in cardiovascular events. So there are these examples that support that a healthy diet is really important in that sense. Regarding the organic, I don't think the field has moved on to that yet. So we know that plants are important. Plants, different fiber, they ferment it in different ways. They give different chemicals. But regarding to organic versus inorganic one versus the novel food, novel proteins that we talk about a lot now, we probably do not know much about those. So wonder whether Emad or Nick has something to supplement.
I agree entirely. I think what Sunny's mentioned about the diversity. The plant is because we can't digest plant cell walls. This is, for us, it's just waste product, but it's actually what they feed on, and it does lead to proliferation of some very healthy ones. I always liken this business about the diversity of microbiota and their feed to, sending, say, 100 people to the desert and asking them to survive. So if they're all lawyers, for example, they'll be pretty useless at setting up a colony. If they're all, teachers, it's same thing. But if there's a skill, a mix, they've got people that can build something, they've got people that can treat people, people that are very strong and physical. You've got to have that diversity of skills and tools, obviously. So there's not much point in having somebody, that's a good builder, but without giving them the tools to build. So that's exactly what happens with the microbiome. You want to feed them things that can lead to, expression of those skills and the tools so that it can work for you. Organic versus inorganic, versus, I'm not really convinced. I think we have enough, studies to prove that it's actually superior or better or whatever it is. It's much more expensive as usual, and it looks awful. But I'm very happy to see if there's any evidence. I'd be the first to be converted, but I haven't really seen anything convincing.
I just want to say our knowledge about diet and the microbiome is very, very limited still for certain food groups, large food groups, like most of what we often eat at McDonald's and KFC and in those packets in the supermarket, all those foods. We have very little idea what we're doing to our bugs. Probably bad things, but we don't know much about it. So it's a big area of active research, and our group and others in Monash and elsewhere are doing work in this, because it's just so important. We have to know, so we can hopefully provide decent advice to people. I also suspect there are going to be differences among people. That's the other key part. Not only are the bugs diverse, we're diverse. And so a diet for one individual may not always be the best diet for another individual. So even if the majority will benefit from a certain diet, there may be some who go the other way. So we've got to work that out as well. More questions than answers. That's science.
I think we have another question from the floor.
Thank you. I might be the first unintelligent question of the night, I'm not sure. But a vision for the future, I'd like to know the panel's idea. When we are thinking about winter weather, people think about having vitamin C. We buy yogurt, that's good for us. What do you see, perhaps this elegant way of having a transfusion, or do you see something in maybe 20 years' time that'll be so off the shelf, so simple? What would you like to see if you don't think you will see it?
What a great question. What a tough question. I'll start because I can think of something off the top of my head. Every baby born will be treated such that their microbiome is maximally beneficial from day dot. Because right now that's not the case. Not at all. There's some efforts to do this actually already, although we don't quite know what we're doing exactly. But I think this will be the future where we maximise people's health for their lifespan by getting in pretty early and doing it safely and effectively. It won't be that hard. So that's one thing that will, I think, change medicine going forward. Not sure about the other stuff, but we'll hear what the others say.
This is the most intelligent question of the night. Absolutely. Honestly, because it's the way for the future. The tragedy here is that we already know that there are certain lifestyle and practices that could prevent 80% of disease. So healthy eating, physical activity or exercise, avoiding all the toxins. All of these things are well known, well established. We've got millions of studies that show that. Yet it's not a strategic objective of governments. There's no government anywhere in the world that would make this the priority, the healthiest nation in the world. Why? Because it doesn't fit with the 4 to 5 year cycle of elections. It's something that you've got to do actually that, over a generation or 2 generations that you can start to see that. But we know that if you eat well, we don't have to have any fancy omics or anything else. Just follow the well-known, healthy lifestyle and make that a strategy. Like this is the obsession of government, incentivizing people, giving tax breaks, making things available, subsidizing healthy food, and taxing the hell out of all the other toxins and stuff. This is something that we can do now without having to wait for the future, but it requires, work at the top. In terms of science, this has never been more exciting. It's just so exciting. Why? Because of the advent of artificial intelligence and the ability to handle masses of data. So when we do a stool analysis, for example, for the microbiome, we generate terabytes of data. This is a huge amount of data. And 10 years ago, we would not be able to actually analyze all of that one. But now, with just the press of a button, you're able to bring together an incredible amount of data to try and see patterns and outcomes and predictive, kind of signatures of certain things. So in the future, and this is not distant, I mean, this is something that's, might happen a lot quicker than 20 years. You would go in, as Nick said, from birth onwards, you'd be able to actually map out what you need to take, what your risk is of certain things, how you can manipulate them. If you go to your GP and take a few samples, maybe able to analyze them and actually put them into all of these kind of AI type stuff and predict, what treatment you need, what side effects you might have, which drugs not to take, which drugs actually be more effective for you. This is all going to happen, and very soon. So it's very exciting, but it can't rely on old people like us. It's got to be the next generation. You've got to actually bring up. This is where the education system and the training is got to actually produce that new army of researchers and people that understand that kind of technology to try and maximize the benefit. But it's all out there for us.
You just have to hope that the chat GPT or the other AI you use doesn't hallucinate when it gets the results. That's all you have to hope, because at the moment it can.
And maybe I'll just add that for me, I wouldn't want to have a poop transplant every day. So definitely it's not something that I would do. And likewise, I wouldn't want to take a bacteriophage or engineer bacteria every day. So I think a lot of it actually for the large proportion of the population, it comes down to the lifestyle. So exercise, the diet. And that's why we have been focusing on the diet. The diet is so important and we understand so little about it. So the diet, at least what we eat as complex as the microbiome, at least. So I think we need a much more better understanding on that. For the FMT, for the phage, I think we still need that because sometimes in specific conditions, in diseases, in cancer, I think it's fine to use those for very specific indication, but for the larger of the population, I think it comes down to the lifestyle.
Okay, thank you. Look, I think we should stop after the most intelligent of the, questions have been asked of the evening. I'm sorry that I am aware that there are a lot more other questions being posted online, but I'm also aware that we have run massively over time, and I'm cognizant of the fact that it's getting late. People will probably want to go home. Now, I want to thank you all for joining us this evening. I hope you had enjoyed it as much as I had. And please also join me to thank our speakers of the evening. All right, thank you, everyone.